all the participants. I, Dr. Jaimini Vyas, again welcome you all on this online faculty development program on life science. Our today's speaker is Dr. K.C. Deshmukh. He is principal at Shri PHG Arts and Science College, Kalol. His topic is Immunology in Current Context. Thank you, sir, for sparing your time and being present at our FDP. We are very pleased to have you. Handed over to you, sir. Thank you.
Good afternoon to every participants. Our today's topic is particularly related with the immunology in current context. Now see, immunology itself is a very rapidly developing field, and it is dealing with the study of various mechanisms, discovery of different mechanisms associated with the immune system. It is also dealing with the development of different types of vaccines. And again, it is also undergoing for recognizing various procedures for manipulation of the immune response, how you can manipulate the immune response and how you can get the maximum benefit for the welfare of human being. Now, if you look for the current context, then current context is a totally different scenario as compared to the historical scenario. If you look in the history, then deadly diseases, they do have occurred during the 17th century and during the 18th century. But then also, unknowingly, the people, they have overcome the diseases. They were believing in some supernatural forces, unknowing of the phenomena which are going on in the body. But they have survived their lives. But today's scenario is totally different. And here, when you are discussing about the immunology, then it is not only the immune system, it is not only the vaccination process, not only the immune response, but the proper operation of all these systems is also a very important aspect. A diverse pathological effects that has been observed due to the immune response. Immune response na karane harmful changes or harmful deadly diseases, they have been developed in the human body. And again, the immune response or the immune system is acting with the non-infectious agents or the non-toxic matter against them. The immune response has been developed. And due to this, we are observing some of the diseases like autoimmunity or rheumatoid arthritis. And again, the total failure of the immune system will be responsible for the development of malignancies, the cancer, or even the rejection of the graft or the exaggerated immune response that may be responsible for the diseases like AIDS, acquired immunodeficiency syndrome, where the deficiency is observed in the immune system, or the exaggerated response when it is observed, then it is causing for the malignancies. So this is a overall immunity or the immune response or a basic concept of immunology. Of course, we will go in detail and we will see with respect to the current context. Today's scenario is along with the coronavirus. The coronavirus is one of the factors that is making you aware of different types of vaccinations, and we are eagerly waiting for the coming of novel vaccines, and the campaign may start during the middle of the January or at the end of January. We may have the process of vaccination, but we are unaware that what may be the different signs. 95% of the success has been observed. But anyhow, let us look at the immunology. In current context, you cannot call it as a current context. It is continuously developing field. Even in the history part, if you look, then there was a disease known as smallpox, and 1 million of the people, they have died. Yes, smallpox caused by the variola vaccine, variola virus, variola major, and it is having a higher mortality rate of 30% of the infants, they are succumbed to death. And in the USA itself, some 1 lakh infants, they cost their lives during the year. And again, 50,000 this type of children, they have been hospitalized in the historical times. But again, we were not knowing about the total defense against this type of phenomena. But vaccination later on came out, and the scientist Edward Jenner, he developed the process of vaccination. He obtained the material from the cow, and it was injected into the body of human being or the child, and the child, it survived the life. And due to this particular vaccination phenomena, today we have totally eradicated the disease of smallpox. In India, in 1975, the disease was totally eradicated, and from the world, in 1980, there is no existence of a disease like smallpox. Only in some African countries, South African countries, there may be few cases that may be observed, but in fact, the virus has been totally eradicated, and we are totally free of this particular disease. Then again, this vaccination phenomena, the different scientists, they have played a very important role. The scientist Louis Pasteur, he has also developed a vaccine for the rabies. See, what I mean to say is, during the 17th century, the vaccination phenomena, it has been started. In the 18th century also, the vaccination was being carried out. And the diseases like smallpox or the diseases like rabies, you can look here for the child, Joseph Mister. He was being vaccinated with the rabies vaccine, and again the material was nothing else but the animal which died due to the rabies disease. The spleen was pulverized, minced, added with some of the chemicals, and that particular fluid that was being injected into the body of that particular individual, Joseph Mister. And this particular young person, he survived of that particular disease. Actually, he was beaten with the rodent animal, with the dog, and he was supposed to die, but he survived his life after the process of vaccination. So this is a stepwise phenomena you can observe in the development of vaccines. Again, if you look for the further inventions and discoveries, see, here we are using the word discovery. There is a difference between the discovery and invention. That discovery is actually the viruses or the immunity or the immune response that is already existing, but we are just removing the cover. And that process, we used to call it as a discover, or removal of the barrier or removal of the lid that we have kept over that particular knowledge, and that is being called as a discovery. It is not being prepared, it is not being synthesized. Of course, it is being detected by the human beings, but it is a discovery which is already existing in the nature. If you look for the inventions, then inventions are something different. There you are talking of the electron microscopes. So during that time, these type of microscopes were not available. But later on, these microscopes, they were being prepared, and those microscopes, they were being used for observing different types of pathogens. But before that, let me show you one of the another slide, and that is also focusing on the immune response or the immunity. Here I have shown the sign of pathogen. This is a pathogenic cell, and this pathogenic cell is being attacked by so many different phagocytic cells. See, phagocytic cells, they are normally present in the human body, in the normal individuals, and they are responsible for degrading or harming the bacteria or the viruses, whatever is there. They are being ingested, they are being lysed, and they are being thrown outside the body along with the circulatory system. So scientist Eli Mechnikov, a Russian scientist, he has observed this particular phenomena in particularly 1893. This has been observed and detected, being discovered, and today we are using this process. We know the phenomena, but during that time it was not known. Immunology was there, but we were not aware. The knowledge was lacking. Immunology is there. But today we are discussing again the immunology in context of current scenario, increasing mortality due to the coronavirus. Again, if you look for the Another picture, this is also focusing on the history part, that Paul Ehrlich, he showed that there are so many different type of cell receptors which are present on the cell. And once the foreign material enters, it combines with this particular cell receptor, and later on it has been observed that there are many number of these cell receptors, they are being produced after the triggering of material from outside. These foreign antigens, they are triggering the cell receptors, and due to that so many different materials, cell receptors, they are being produced. Actually this particular material, a cell receptor, actually it is nothing but these are the antibodies, and these antibodies, they have been attached over the surface of B cells, and these B cells, they are being triggered after the attachment of the antigen, after the attachment of foreign pathogen to the cell receptor. So this cell receptor itself is an antibody, and after this particular attachment, the cell is being triggered to produce many number of these antibodies. So this is also a immunological process. It has happened a decade, 100 years back. This has been detected. The immunology was present during that time also. But again, we are considering it. Again, it has become a need of time to focus on the historical aspects. After the discovery of the electron microscope, we are in a capacity, we are in a scenario, we are in an environment to detect the rotaviruses. Rotaviruses, they are also responsible for the death of so many infants, infantile diarrhea. Again here, the phagocytic cells, they are engraving, they are ingesting the bacilli, the organisms. They are also responsible for the diseases. So this is the electron micrographs. The point is just to show you that how they are appearing and how they can be detected. This is a today's scenario. We are having the all latest instruments or the different techniques. See, this is a yeast cell, a yeast Candida albicans. See, this particular yeast is responsible for the disease in the mouth, in the vaginitis, or the intestinal ulceration. So this is a particular electron micrograph or electronic picture. You can observe that these are the non-visible things, but they are why they are responsible. They have a specific shape and arrangement, and they are causing different type of diseases. Here, once again, a nematode that has been attacked by the phagocytic cells, and it was totally destroyed. So here you can see that electron micrograph, it is also helping you in detecting and analyzing the different type of pathogens which are responsible for causing different diseases. Whereas a current scenario or a current concept, what is a current immunology? Current immunology is totally different. During that time, we were unaware of the B cells, not aware of the phagocytosis. They were unknown phenomena. But today, after passing these milestones of the history, today we know totally very well about this phenomena, antigen antibody reactions or the detection of the antigens by using the antibodies. And again, these antibodies are again detected by anti-antibodies which are connected with the enzymes where you are adding the substrate and you are getting the colorful reaction. The tagged reagents, antigen antibody reactions, and due to that you can detect many different type of diseases. You can further categorize these reactions, that precipitation reactions or the agglutination reaction that can be detected. Here the other picture, a syringe showing you the process of vaccination. See, the process of vaccination is very, very old, but today it has been changed. The phenomena or the process or the technique or the involvement of the genetic engineering is giving you the current context. It has been totally changed. If you look for the below picture, this is a process of production of monoclonal antibodies. See, these are the antibodies, these are being produced in a very, very large amount, and these are produced in vitro process. Normal disease ni andar antibody production sharir ni andar thatu hoy, but these antibodies are very limited. These antibodies are used for the detection of antigens or the detection of pathogens. But here, these antibodies are produced in a large amount. How they are produced in large amount? That you are taking the spleen cells which are already injected with the antigen. Any જોડે તમે mix કરો cancerous cell છે mouse નો, and you are getting the hybrid cell, a cell from the spleen and a cancerous cell or myeloma cell from the mouse. If both these type of cells are mixed, you are getting a hybridoma, and this hybridoma, when it is grown in a het medium, hypoxanthine, aminopterin, thymidine medium, then you are having a clone of hybridoma cells. They have been grown nicely, and further you can have the production of antibodies from these cells. It has the character of continuously growing from the cancerous cells, and it has the capacity to produce only one type of antibody from the spleen cell. They are injected with only one type of antigen. So production of monoclonal antibodies, they are being used on a very, very large scale for detection of different type of analysis, blood grouping process, or even in the pregnancy test, or even in the passive immunity. આ બહુ જ મોટા પ્રમાણમાં આનો ઉપયોગ બધો કરવામાં આવે છે. So this is the current immunology, the advancement in the immunology that has been proved as a boon for the human welfare. Further again, if you look, that it is the only solution. What is actually immunology? You can come to the conclusion that immunology is the only solution for EID. What is the meaning of EID? Emerging infectious diseases. How these infectious diseases are coming out? Do the pathogens are hidden? Do they are not exposed to the human being? Yes, they are hidden in the environment, they are hidden in the nature, they are hidden in the animals, and these animals are not being normally exposed to individual, and that is why human being as not coming across these animals, we are protected. But here you can again see that human welfare is only secured through science and study of immunology. Just now we have focused on the historical aspect, that how the immunology is playing a important useful role. Here you can see that human welfare is only secured through science and study of immunology. Again, if you look further, what is the current context? Current context is related with the sufferings of human being, maybe at the cost of life. કરંટ કન્ટેક્સ્ટ છે એટલો ભયંકર છે કે જ્યાં હ્યુમન બીંગ તો સફર થાય અને ઘણી વખત એનો જીવ પણ એમાં એને ગુમાવવો પડે. અને એનું કારણ શું છે? ઇમર્જન્સ ઓફ ઇન્ફેક્શિયસ ડિઝીઝીસ. અને આ કોનું છે? પર્ટિક્યુલરલી you can talk about the coronavirus. And again, what are the factors? You can see the Chinese, what they have done. They have used some of the rodents for eating purpose. Changes in nutritional habitats in search of novel food, killing rodents and other animals for eating purpose. Due to that, the virus has been transmitted from these rodents or these particular animals to the human being. And further, human-to-human transmission has occurred. Second thing, if you look, that modern transportation facilities. Virus once comes to the human being, it may be transmitted very easily or very faster to the modern transportation facilities. People traveling from foreign countries to India, or some people from China or from other countries. Throughout the globe, people, they are traveling. And due to this travel, the virus was highly spreaded. Initially, it was endemic. Endemic એટલે અમુક જગ્યા પૂરતો મર્યાદિત હતો. Later on, it has spread to the community, epidemic, and later on, throughout the globe, throughout the world, the virus spreaded. જેને આપણે મહામારી કહીએ છીએ કે પેન્ડેમિકના સ્વરૂપે આ વાયરસ આખો ફેલાઈ ગયો. હવે આની અંદર once the virus has been spread, we are not having proper public health measures, or we are not following public health measures. We are not obeying the government regularity rules, and that is why we are having the problem. Widespread use of, why again the emerging diseases? એનું બીજું કારણ આ પણ છે કે widespread use of antibiotics, third generation antibiotics કે જે બહુ જ high dose ની અંદર કે જેની effect બહુ જ વધારે હોય, use of those antibiotics, or again the use of pesticides in the agricultural crops. These are responsible for the development of resistant microbial flora. એવા પ્રકારનો microbial flora કે જેની ઉપર કોઈ પણ એન્ટીબાયોટિકની અસર ન થાય અને એને નાથવું કે એનાથી બહાર આવવું કે એનાથી રોગ ન થવો દેવો કે just to come out of that particular infection has become difficult. Global warming and unexpected seasonal changes leading to more reservoirs and spread. Here the reservoir is indicating to the ecological needs or the various different habitats, unexpected seasonal changes થવાના કારણે એવા એવા વાતાવરણનું નિર્માણ થાય છે કે જ્યાં ગાડી એવા ઓર્ગેનિઝમ ગ્રો થાય કે જે આજ દિન સુધી આપણે જોયા ન હોય કે એ વાતાવરણની અંદર ક્યારે પણ પ્રવેશ્યા ન હોય, but we are providing them the chance for coming out of that ecological mix, coming out from the animals, coming out from the rodents and spread into the community. Again, what are the principles? Immunology એનો મુખ્ય આધાર શું છે, એના સિદ્ધાંતો, પાયાના સિદ્ધાંતો શું છે? તો ઇમ્યુનોલોજી એ જુદા જુદા પ્રકારની હોય જેને આપણે ઇમ્યુનિટી કહીએ છીએ, અને ઇમ્યુનિટીના પ્રકાર છે. Immunity is of two different types, innate immunity and acquired immunity. See, innate immunity itself is a nature's gift, or it is the God's gift, that we are protecting our body against the attack of different pathogens or attack of different organisms from the external environment. It is a boon, or it is a God's gift that along with the birth ની જોડે જોડે આ innate immunity આપણને મળેલી હોય. એના ઉદાહરણો આપવાના થાય તો we are having a skin surface layer, we are having the hairs, we are having the nails. These are protecting our body from the attack of pathogens from the outside. બહારના pathogens શરીર ઉપર અટેક કરી શકતા નથી. Even inside the body, if you look, we have a mucus secretion. લાળ સતત મોઢાની અંદર ફર્યા કરે છે. See, that is not allowing any external virus or bacteria to stick inside the mouth, to stick in over the teeth and further proliferate or further grow. એ grow થવા દેતું નથી. Innate immunity is found to be responsible. પછી, if you go inside further, if you go in the blood, then again the blood, it has so many different type of cells, phagocytic cells. It has different type of enzymes. It has different type of chemicals. It has different type of cytokines or interleukins that will not allow the pathogen which has entered to survive. So this is an innate immunity. It is a birth-born immunity. Along with the birth, we are taking birth along with the innate immunity. This immunity, innate immunity, is found to be non-specific. Skin is not allowing any type of pathogen to enter. Mucous membrane is not allowing any type of pathogen to go inside. So this is not specific. Irrespective of the any pathogen, we are having the protection. The other one is the acquired immunity. What do you mean by acquired immunity? See, acquired immunity itself is acquired only after the birth. It is not God's gift, but of course acquired immunity is also a gift in another sense, you can say. But the only thing is, it is acquired after the birth. As the individual come across different type of pathogens, different type of chemicals, different type of food, the acquired immunity, it is being acquired from the environment. જેવા ઓર્ગેનિઝમ્સના સંપર્કમાં આપણે આવીશું એ પ્રકારના એન્ટીબોડી શરીરની અંદર ડેવલપ થશે. અને again you can say that there are two different types. One is the natural and other is the artificial. કુદરતી રીતે સંપર્કની અંદર આવ્યા. Naturally an individual is suffering with the disease, then naturally the immune system will be activated and naturally we will have the antibody production. હવે તમે બીજું જુઓ કે it may be artificial. Acquired immunity develop થઈ શકે, artificially પણ activate થઈ શકે. Okay, once we are going for the process of vaccination, artificially you can have the production of antibody. એટલે કુદરતી રીતે અટેક થાય કાં તો પછી વેક્સિન્સ દ્વારા શરીરમાં ઓર્ગેનિઝમને તમે ઇન્ટ્રોડ્યુસ કરો, બંને રીતે તમારા શરીરની અંદર ઇમ્યુનિટીનું પ્રોડક્શન થાય છે. Again, this acquired immunity, it is found to be natural or artificial. Further again, you classify it. That acquired immunity may be further developed either actively or passively. See, the active word indicates the activity or the role or the activation of the immune system. Acquired immunity કુદરતી રીતે મળી હોય કે આર્ટિફિશિયલ રીતે મળી હોય, એની અંદર જો immune system ને એક્ટિવેટ કરવામાં આવે so that becomes actively acquired immunity. Maybe natural or maybe artificial. But once again, if that particular immune system is not activated, but then also you are having the antibodies. Naturally you are having the antibodies. Individual is having the antibodies being acquired artificially. So here you can give the example of the infants or small children or the infants who are particularly being fed by the mother's milk. It is having a large portion of the yellow color cholesterol milk, a large amount of antibodies, a large amount of protein, which is protecting the children, protecting the child against different type of diseases. This is the passive. The antibodies are being taken, being provided to the child by the mother. But again, if you look, then artificially these prepared antibodies can also be injected into the individual, particularly in the cancerous patients or the AIDS patient. You can introduce these antibodies. So passively, it is possible to undergo for different types of immunity that can be developed in the body. Again, once talking about the immunity, there are immune responses. Immune responses, again, this is a phenomena where the body is responding to the foreign material. બહારથી આવનાર કોઈ પણ વસ્તુ, કોઈ પણ સબસ્ટન્સ, કોઈ પણ પદાર્થ, any particular biomolecule or molecule, any type of substance which is not involved in the composition of the body, chemical composition of the body, does not possess that particular composition which is coming from outside. એને આપણે કહીએ છીએ foreign પદાર્થ, foreign substance. હવે એવા બધા પદાર્થની સામે, એવા બધા બાયોમોલિક્યુલ્સની સામે we are developing the response, and that we are calling it as a immune response. એના characters શું છે? આ immune response જે છે એ immune system produce કરે છે, પણ immune system જે છે એ discriminate કરી શકે છે. Immune system can discriminate, it can see, it can identify whether the foreign molecule has coming is different or it is matching to the body. If the molecule is matching to the body, then immune system will not produce the immune response. If that foreign molecule is different, then immune system will produce the antibody. Otherwise, it will not produce. So it has the power of discrimination. Immune system identify કરી શકે છે. Immune system ની અંદર diversity પણ ધરાવે છે. Diversity એટલે different types of antibodies can be produced against different types of foreign molecules. જુદા જુદા કેટલા પણ ભલે ને એની અંદર શરીરની અંદર પેથોજન્સ આવી શકે during the lifespan, દરેકની સામે antibody produce કરવાની ક્ષમતા immune system ધરાવે છે. See, this is a natural phenomena that individual can produce a diversified different types of antibody molecules during the lifespan. The third thing is the specificity. What do you mean by specificity? Okay, whatever the antibodies are produced, they are found to be highly specific to the substance which are responsible for their production. જે foreign પદાર્થ એના production માટે જવાબદાર હોય, એની જોડે જ ફક્ત આ એન્ટીબોડી જોડાશે અને એને ન્યુટ્રલાઇઝ કરશે કે એ પદાર્થને શરીરમાંથી બહાર કરી દેશે. So this is the character. આ જે રિસ્પોન્સ તમને મળે છે એને આપણે ઇમ્યુન રિસ્પોન્સ કહીએ છીએ, અને this particular immune response is being provided by the immune system. Then again you have the memory. Immune system has the memory. કે immune response ને આપણે કહી શકાય કે memory છે. Immune response એક વખત ડેવલપ થયો અને એને જ ફોરેન પદાર્થ ફરીથી શરીરની અંદર દાખલ થાય. If the same biomolecule is entering into the body, then body will produce the exaggerated immune response or the excess antibody production. પહેલા જે antibody production થયું હશે કે પહેલા જે immune response થયો હશે એના કરતા વધુ પ્રમાણમાં અને બહુ short time interval ની અંદર તમે antibody નું production થયેલું તમે જોઈ શકશો એમાં. So immune response આ character છે. એટલે કોઈ વ્યક્તિને એક વખત ટાઇફોઇડ થયેલો હોય તો એના લાઈફ સ્પાન દરમિયાન આવનાર 6 થી 7 વર્ષ દરમિયાન મિનિમમ એને ટાઇફોઇડનો ફરીથી હુમલો થતો નથી કે ફરીથી એ વ્યક્તિને ટાઇફોઇડ થતો નથી. So this is a very important phenomena that immune response has the character to memorize, and that pathogen, that particle is again not being allowed to propagate or multiply inside the body once again. એટલે આવા જે foreign molecules છે કે આવા જે બહારથી આવનારા માઇક્રો ઓર્ગેનિઝમ્સ છે કે બાયોમોલિક્યુલ્સ છે એને આપણે સંબોધન કરીએ છીએ કે they are being known as antigens, અને જે રિસ્પોન્સ એની સામે તૈયાર થાય છે એને આપણે કહીએ છીએ એન્ટીબોડી રિસ્પોન્સ. હવે આવા એન્ટીબોડી પ્રોડ્યુસ કરવા વાળા જે કોષો છે કે જેને બી સેલ આપણે કહીએ છીએ, ઇટ ઇઝ રિસ્પોન્સિબલ ફોર પ્રોડક્શન ઓફ એન્ટીબોડી. It can be transferred. એને આપણે ટ્રાન્સફર કરી શકો છો. You can transfer the B cells from one body to another body. Passive immunity તો છે જ કે already ready-made antibodies, they can be injected into the other individuals for the purpose of protection, for the purpose of defense. હવે આ antigens શું છે? What are the antigens? See, these are some of the terminology which are being used commonly. We are acquainted with reading our newspapers. Particularly, we are not frequently going for the English language, but so many scientific terms they are being used in the English newspapers. Particularly the terms like pandemics, epidemics, or clinical trials, or the antibodies, or the vaccination. આ બધું બહુ જ commonly એની અંદર બધું ઉપયોગમાં લેવાય છે કે વેક્સિન્સના જુદા જુદા નામો કે એટીન્યુએશન આ અમુક શબ્દો છે. They are very commonly being used in the English newspapers. If you are having the habit of reading that, then every day we can have some news related with the immunology, or related with the infections, or related with the microbiology, or related with the biotechnology. So here the antigens, see the word itself, anti and gens. If you split the word antigens, so they are generating, what they are generating? They are generating antibodies. Antigens, they are responsible for generating the antibodies. And what are this actually? And chemical nature શું છે? કે they are foreign chemicals. They are the chemicals, they are coming from outside. Coming from outside means they should not match with the chemicals which are existing in our body. બહારથી આવે એનો પ્રશ્ન નથી, પણ શરીરની અંદર રહેલા જે બાયોકેમિકલ્સ છે એની જોડે જો ફોરેન કેમિકલ્સ મેચ થતા હશે તો that substance will not work as an antigen. So antigen should be totally foreign. Foreign means it should not match with the biochemical composition or with the chemicals which are existing in our body. So this is very important. So here again one statement is being made that all the pathogens are antigenic, but all antigens are not pathogenic. What is the meaning of this? જેટલા પણ જીવાણુઓ વિષાણુઓ છે એ બધા એન્ટીજેનિક છે, એ બધા જ એન્ટીબોડીનું પ્રોડક્શન કરી શકે છે. Even you can look for today's scenario that due to the coronavirus, many people they are found to be asymptomatic, or they are not found to suffer with the coronavirus. So this particular coronavirus, it has functioned as a pathogen, but it is antigenic, and it has developed the antibodies in the healthy individuals, and they have not suffered with the disease. But again, the contradictory statement, but all antigens are not pathogenic. Pathogens બધા એન્ટીજનનું કામ કરે છે, પણ બધા એન્ટીજન્સ જે હોય, જે એન્ટીબોડીઝ પ્રોડ્યુસ કરતા હોય, એ બધા પેથોજન હોય, એ રોગ કરતા હોય, એ જરૂરી નથી. So what is the example of this? Antigens are not pathogenic. આ તમને ક્યાંથી મળે? વેક્સિન્સ માંથી મળે. વેક્સિન્સ ને શરીરમાં દાખલ કરો તો એન્ટીબોડીનું પ્રોડક્શન તો થાય, એ એન્ટીજનનું કામ તો કરે, એન્ટીબોડીનું ડેવલપમેન્ટ તો કરે, but they are not responsible for causing the disease. So this is a basic concept of immunology. Antigens, they are producing the antibodies. એ ગ્લોબ્યુલર પ્રકારના પ્રોટીન છે અને ન્યુટ્રલાઇઝ કરે છે એન્ટીજન્સને, પેથોજન્સને એનું ઓપ્સોનાઇઝેશન કરે છે. ઓપ્સોનાઇઝેશન એટલે એ જે પેથોજન હોય એનું સેન્સિટાઇઝેશન કરે, અને એવા સેન્સિટાઇઝ થયેલા પેથોજન્સ છે એ બહુ ઇઝીલી ફેગોસિટાઇઝ થઈ જાય. હવે જુદા જુદા પ્રકારના એન્ટીબોડીઝ તમને જોવા મળે. IgM, IgG, IgA, D and E. 5 different types of immunoglobulin molecules they are being observed. And again, these are ultimate globular proteins responsible for providing the defense mechanism. એના કારણે આપણે ડિફેન્સ મિકેનિઝમ મળે છે, શરીરની અંદર પ્રતિકાર શક્તિ થયેલી જે જોવા મળે છે. This resistance, or particularly we are calling it as a defense, is nothing but the immune response or the immunity shown by the individual. હવે નીચે અહીંયા જે સેપરેટ કરેલું છે મોનોક્લોનલ એન્ટીબોડીઝ. These are only a single type of antibodies, and they are having a vast variety of different uses. આપણે પહેલા જ એને જોઈ ગયા, પણ એન્ટીબોડીનું ટાઇટલ છે એટલે આ પણ એક મોનોક્લોનલ એન્ટીબોડી. ઇન વિટ્રો પ્રોડક્શન ઓફ કોર્સ એનું થતું હોય, but they are a different type. Of course they are antibodies. હવે અહીંયા ઇમ્યુનિટી જે બે પ્રકારની તમને જોવા મળે છે, હ્યુમોરલ અને સેલ મેડિએટેડ. હ્યુમોરલની અંદર એન્ટીબોડીનું પ્રોડક્શન થાય. આ જેટલા પણ છે આ બધા પેથોજન્સ છે, ફોરેન પ્રોટીન્સ કે પછી વાયરસીસ કે પછી બેક્ટેરિયા કે પેરાસાઇટ, આ બધા જ્યારે પણ શરીરની અંદર દાખલ થાય છે તો they are being engulfed, they are being phagocytized, અને જે પણ કોષો છે અહીંયા આગળ ડાયજેસ્ટ કરે છે. આ પેથોજન્સને ડાયજેસ્ટ કરવામાં આવે છે અને એન્ટીજન સ્વરૂપે એને ટી હેલ્પર સેલ એને આઈડેન્ટિફાય કરે છે. અને આવા ટી હેલ્પર સેલ્સ they are activated after coming in contact with this pathogen. Our T helper cells B cells ને activate કરે છે અને આવા activated B cells antibody નું production કરે છે. હવે અહીંયા ઘડી તમે જુઓ કે B cells they are responsible. T helper cell નું અહીંયા એક કામ થઈ ગયું. બીજું અહીંયા T C cells T પ્રકારના બીજા કોષો છે T cytotoxic cells or T cytolytic cells, they are also identifying this antigen. And again they are being activating by secreting the chemicals. Once again they are activating the B cells for the production of antibody molecules. So this is a process in which the foreign pathogens or the foreign antigens, they are being ingested, they are being exposed outside to the B cells directly, or they may be exposed to the T cells. And B cell exposure માટે T helper cells એ અગત્યનો રોલ ભજવે છે. એ એન્ટીજન જોડે કમ્બાઈન થઈને એને એક્સપોઝ કરે છે, એક્ટિવેટ થયા પછી એને એક્સપોઝ કરે છે. આ ઇન્ટરલ્યુકિન, આ જુદા જુદા કેમિકલ પદાર્થો રિલીઝ થાય and they are responsible for the activation of the B cell. B cells are converted into plasma cells and they are actually producing the antibodies. If you look for the T cytolytic cells, then again these cytolytic cells or the cytotoxic cells, they are also producing the interleukin or chemical mediators. They are also activating the B cells which are responsible for the antibody production. Or a cytolytic T cells share, they can also directly destroy the foreign pathogens. So this is particularly, you can say there are two type of mechanisms. એક તો antibody નું production થાય છે જેને આપણે humoral કહીએ છીએ, or there is a cell mediated immune response where the T cells are playing a very very important role. So these are the basic principles. Now if you look for the applied areas of immunology, what are the different applied areas? You can look here, the use of immunological reactions. જુદા જુદા પ્રકારના reactions કરાવવામાં આવે. એમાં antigen can combine with the antibody. જો આપણી જોડે antigen હોય તો you can detect the antibody. એનું example આપવાનું થાય તો માણસના શરીરની અંદર જો typhoid થયેલો હોય, if the individual human being is suffering with the typhoid disease, then naturally we produce the antibodies, and these antibodies will be detected by the antigens. Antigens દ્વારા માણસના શરીરમાં રહેલા antibodies નું detection તમે કરી શકો. You know that what are the type of antigens you have. You are having the antigens against the typhoid disease. So these antigens, they combine with the antibodies. કારણ કે immune response, they are highly specific. Typhoid na antibody હોય તો એ typhoid na antigen જોડે જ combine થાય. એટલે અહીંયા you are looking for this antigen antibody reaction. But here we are using the word tagged agents. What do you mean by tagged agents? Tagging એટલે બાંધવું. We are binding. You can bind either the antigen or you can bind the antibodies. You can bind with the enzymes, you can bind with the radioisotope, you can bind with the fluorescent dye. Rhodamine or auramine, જુદા જુદા પ્રકારની dye આવે, fluorescent dye. They will go for a emission of longer wavelength light if they are focused with the ultraviolet radiation. See, longer wavelength light, a different type of glow or fluorescence that can be very easily detected. એટલે આ બધા tagging agents છે that can be bound either with the antigens or they can be bound with the antibodies, and they can be used respectively for the detection of antigens or antibodies. Or you can also go for binding this antigen or antibody with the enzymes. And a bind antigen antibody હોય એની અંદર substrate નાખવામાં આવે. If you are reacting with the substrate, then you can observe for the colorful reactions. So detection of many different type of diseases. આમાં તમને antigen antibody precipitation reactions તમને જોવા મળે, agglutination પ્રકારના reactions તમને જોવા મળે, and they are having a widespread use in the forensic laboratories or for the detection of different type of diseases or the detection of Ascolis test. જેને આપણે anthrax પ્રકારનો જે રોગ છે એને find out કરવું હોય in the animals, particularly in the cattle. Once the disease is spreading, then it is spreading very widely, developing the epidemic and responsible for death of many animals. So you can detect these type of different diseases by using the antigens or antibodies. You have the production of monoclonal antibodies. Therapy માટે you can use જુદા જુદા પ્રકારની કેન્સર થેરાપી or કોઈપણ પ્રકારનો રોગ થયેલો હોય તો એની અંદર directly you can provide that individual with the antibody molecules. You can go for the treatment of AIDS, even the treatment of corona patients, passive immunization, or you must have read about the plasma donors. બ્લડ નથી લેતા પણ પ્લાઝમા લે છે. રેડ બ્લડ સેલ્સ માંથી કાઢી નાખવામાં આવે અને જે વધારાનું ફ્લુઇડ છે which is having the antibodies, they are provided with they are being used to transfusing the patients who are suffering with the corona disease. Passive immunity, એટલે આ એક પેસિવ ઇમ્યુનિટી છે એની અંદર પણ ડાયરેક્ટલી રેડીમેડ એન્ટીબોડીઝનો આપણે ઉપયોગ કરી શકાય. ઓર્ગન ટ્રાન્સપ્લાન્ટેશન, જુદા જુદા પ્રકારના ગ્રાફ શરીરની અંદર સર્વાઈવ થતા નથી કારણ કે ફોરેન છે, બહારથી આવેલા છે. unless and until they are matching, they cannot be accepted by the body. Twins હોય અને એકની અંદર એક કિડનીનો પ્રોબ્લેમ હોય તો બીજા બાળકમાંથી બે જોડકા હોય તો એકમાંથી આપણે એને ટ્રાન્સપ્લાન્ટ કરી શકાય બીજા બાળકની અંદર કારણ કે એની અંદર જેનેટિક મેચિંગ છે, એની અંદર એન્ટીજન્સનું મેચિંગ છે. હવે આ જે એન્ટીજન્સ છે કે જે ટીશ્યુ ઉપર કે ઓર્ગન ઉપર જોવા મળે છે એને આપણે કહીએ છીએ that antigens we used to call it as a MHC antigens. એમાં બે પ્રકારના એન્ટીજન્સ છે. MHC1 they are found in all the nucleated cells અને બીજા છે MHC2 કે જે તમને ઇમ્યુન સિસ્ટમની અંદર ઇન્વોલ્વ થયેલા જે કોષો છે, જે બી સેલ્સ છે, ટી સેલ્સ છે કે ફેગોસાઇટિક સેલ્સ છે, એની અંદર MHC2 પ્રકારના એન્ટીજન્સ તમને જોવા મળે છે. Once you are transplanting the graft, you may have the host versus graft. Host in which you are implanting, you are transplanting the graft. Host may not accept or may accept depending upon the MHC matching. This is one reaction, host versus graft, or the graft itself, if they are having the activated cells, then they may produce the antibodies or they may release the T cells, activated T cells, and they are having the damaging effect in the host body. So this is particularly a role played by MHC, and MHC is again, today we are having at ease about the knowledge of this different type of antigens and antibodies, and that is why the successful transplantations are being possible. Now let us come to the topic of immunoprophylaxis. What is the phenomena? What is the prophylaxis? Prophylaxis is treatment in advance or providing the protection before the development of disease, and it is possible particularly with the immunology by using the different type of vaccines. What are the different types? Attenuated whole agent vaccines. Attenuated કે જ્યાં ઘડી bacteria કે virus, they are used as a vaccine, but their virulence power has been decreased or it has been totally removed. And આની અંદર once the disease producing capacity has been removed, then they can be used as a vaccine. They can be directly injected into the body as they are attenuated and whole agent, they are living. They can undergo for further multiplication throughout the lifespan. They can remain in the body and we can have the continuously total active immunity throughout the life. Inactivated whole agent, where the bacteria or the viruses, they are inactivated, they are in the dead form, they are used, but primary, secondary or tertiary doses of such type of vaccinations are required. Then toxoids, different type of toxoids, diphtheria na toxoid, toxin છે કે પછી pertussis પ્રકારનો જે રોગ છે કે tetanus નો જે રોગ છે એ બધા toxins એને તમે ટ્રીટમેન્ટ આપો, chemical treatment, heat treatment, radiation treatment, they are converted to toxoids and they are used for vaccination. Subunit vaccine, આખા virus કે bacteria, they are not used, but the fragment, it is responsible for antigenic stimulus, they are used. Conjugated vaccines, you are combining one particular antigenic fragment with another chemical substance like proteins, polysaccharides and proteins, they are combined together for immunization process. And the last one is the nucleic acid vaccine. See, this particular nucleic acid vaccines, they are found to be the most modern concept of utilization of vaccination. They may be of different types. Deoxyribonucleic acid, RNA type of vaccines, they may be used for directly injecting, directly introducing into the muscle cells, and these muscle cells further will go for the production of antibody molecules. Now see, coming after knowing the basic aspects of this immunology, you can look for the type of COVID-19 vaccines which are supposed to be produced or which are in the production process, which are in the pipeline, and they will be produced in a very short time interval. So there are different type of nucleic acid vaccines. Just now we have seen that this genetic engineering concept has made it possible. DNA vaccines, company in vivo, and gene that codes for the virus protein that is injected into the individuals. Then again we are having the RNA vaccine, we are having the viral vector vaccines. See, here it is something different, that the viruses, coronavirus, DNA, genes, they are introduced into the another virus. The virus like adenovirus, any other corona genes તમે દાખલ કરો. Adenovirus is not responsible for causing any particular damage or injury or any type of infection. And then if you are introducing this virus into the body, the DNA or the RNA will come out and will enter into the muscle cells. They will acquire this particular genes of the virus and they will produce the immune response, particularly activation of B cells and activation of the T cells. RNA-induced antibody production, RNA vaccines, protein vaccines, that you are not using the RNA or DNA, but you are taking the protein component or the protein fragment. See, this is a totally a foreign molecule or foreign antigen. And when you introduced into the body, it is responsible for the antibody production. Virus subunit vaccine, કોઈ પણ તમે spike protein કે nucleocapsid protein તમે લઈ શકો, or it has been taken and it has been used for the process of vaccination. So there are different companies which are using this. These are the procedures which are being undergone. These are the very complicated procedures. But talking, reading and understanding is simple and very easy task. But preparation of such type of vaccine, it is found to be highly difficult. But it is in the going on process. See how it is being prepared or what are the type of vaccines. This is a central dogma of biology. કે જે character DNA ની અંદર હોય એ RNA માં ટ્રાન્સફર થાય અને that character represented in the form of protein. The type of genes is responsible for the type of protein. Transcription, translation, RNA, and RNA will produce the protein. So this is a basic central dogma of biology which is being used for the preparation of different types of vaccines against the coronavirus. See, this is a DNA vaccine. It is being injected into the body. DNA is injected into the body. It will produce RNA. RNA will produce the protein. This protein is nothing but they are the type of antibodies which are providing the protection. In Pramana directly you can use the RNA for the vaccination process. The time period can be reduced or the time span can be reduced by directly providing the RNA vaccines. And still you can reduce the time period by directly providing the protein vaccines, nucleocapsid or the spike proteins. See, the SARS-CoV outbreak has prompted the rapid expansion of several technology platforms, including DNA and RNA vaccines. Never before clinically tested in humans. હવે અહીંયા જોવા મળે છે કે 95% success is being observed or 98% success or 90% success. Some people are having the sufferings and majority of the mass they are protecting themselves against the virus. But this is જે 17મી સદીની અંદર જે આપણે ચિકન પોક્સની વાત કરી કે વેક્સિનેશનની વાત કરેલી, the same scenario has going on in today's circumstances. So immunology in current context is again become live or it is already live. It is going on thoroughly from historical time till today. You cannot call it as a history because it was already existing. It was discovered. It was not invented. It was discovered. It was already existing. Then again, DNA or RNA vaccines direct our cells to make parts of viral protein that do not cause disease but provoke the immune system. DNA અને RNA દ્વારા પ્રોટીનનું નિર્માણ થાય અને આ પ્રોટીન્સ જે એન્ટીજનનું કામ કરે એના દ્વારા એન્ટીબોડીનું પ્રોડક્શન થાય. Immune system will produce the antibodies. So just such type of protein can be directly injected into the body. Today, rapid production of DNA or RNA in large amount only requires the sequences of viral genetic material. SARS-CoV virus 2 was identified and published by the Chinese researchers on January 11, 2020. Recently, the process has been going on in the last 6 months. Also, other novel vaccines are being developed like protein subunit vaccine, split virus vaccine. There is also development of synthetic substances called the virus-like particles, which are matching structurally with the viruses. They are responsible for mRNA production and antibody production. So instead of antigens, virus-like particles, they are used. See, this is the transfer, vector vaccine. This is a coronavirus. In our DNA ke RNA, it તમે transfer કરો છો. કોની અંદર you are transferring it to the harmless virus. This harmless virus will have the DNA from coronavirus. And now, this will go for injection into the body of human being. Viral vector vaccines are similar to live attenuated vaccines in that they use a harmless virus known as vector to carry a gene encoding the antigen of interest. When the vector virus infects a cell, they administer this foreign gene into the cell. The cell then transcribes and translates the genes into the production of antigen to produce the antigen S protein, which is displayed on the cell surface to stimulate antibody production. Now there are various companies. The infected cell may also slowly reproduce the virus, which allows more cells to become infected and produce more antigen, thus amplifying the effect. Infected cell એનું multiplication સતત થવાનું છે because it is living and the effect of exposure of the antigen is amplified, it is multiplied, and as long as the antigen is exposed to the immune system, there will be the production of antibody. See the companies Oxford, AstraZeneca, Gamaleya, Sputnik, and the Janssen vaccines are all vector vaccines. They use a harmless endovirus as a vector for gene therapy. See, these are the variety of different types of COVID-19 vaccines, DNA vaccines, and the name of companies that I have shown. DNA vaccine, RNA vaccine, viral vector, viral subunit, and the Novavax, AdaptVac, Clover Biopharma. See, these are the different companies which are responsible. They are actually undergoing for the clinical trials, and within 2 months, many such vectors will be made available in India itself at 3 places. One at the Serum Institute in Pune and Hyderabad, and one in the Cadila laboratory in Ahmedabad. They are preparing these vaccines for the coronavirus. Potential challenges in the immune system development. See, we have seen for the immune system. But what is an effective immune system? Effective immune system તમે કોને કહેશો? What are the characters? An effective immune system must interpret the changes which are going on in the world. Respond properly. જેટલા પણ changes થતા હોય એના માટે એ તૈયાર થાય. It overcome the challenges. It should work in the different environment with different pathogens. The concept is different pathogens, their exposure, the immune system should be ready to produce the response at all the time. It should have the ability to adapt to strange environment. વાતાવરણમાં ભલે ને બદલાવ થાય, global warming થાય કે seasonal changes થાય, it should detect. A healthy immune system has a symbiotic microbial farm. What is the meaning of this? Immune system ની અંદર શરીરને ઉપયોગ કરતા microorganisms ની હાજરી હોવી જોઈએ. In the previous lecture, we have seen that microbiome, we have heard. And this microbiome, it should be helping the human body. Then naturally the immune system will be lifted, immune system will become powerful. Harmful reaction બહુ ઝડપથી જોવા મળશે નહીં. See, a different door is used for every infection, and blocking this route can stop the infection. જુદા જુદા pathogens જુદા જુદા રસ્તેથી શરીરની અંદર દાખલ થતા હોય છે. The immune system neutralizes an infection. But that pathogens, they are being neutralized by producing the antibodies. But this should occur at proper time. અમુક સમય, within a limit, the pathogens must be stopped. Their multiplication must be stopped. Otherwise, once they have increased the infective dose, then they will be responsible for the disease development. Before their proliferation, before increasing in the number, they should be stopped. We must stop the infection before it is established in the body. Suggested food and supplementary for the immune system. Healthy food હોવું જોઈએ. Healthy food and hydration are vital. Healthy food and hydration. Proper fluid should be taken. These are the vital things. Making an individual living. Liveliness will be observed only after the healthy food and hydrations. Vitamins and minerals. Food should have the vitamins and minerals. Vitamin B, which is insoluble in water, it protects from the infection. Vitamin C protect from flu-like symptoms. Insufficient vitamin D and E. Suppose oil seeds and fruits, if they are not being taken, then naturally one will not be having the sufficient amount of vitamin E, and they may be leading to more chances of attack by the coronavirus. Insufficient and excess iron can also lead to infection. Iron should be in an optimal amount. It should not be excess, it should not be less. Zinc, again the microelement, it should be required in proper amount for maintaining the immune system. And the last one is the food rich in protein is the top priority. Why food rich in protein is a top priority? Because antibodies, they are made up of proteins. And these proteins, ultimately, of course the body is synthesizing, but these proteins are lifting the antibody production. So boosting the immune system is a potential resource for the treatment of patients with the COVID-19, and diet, physical activity and proper sleep must be there. There is no registered medicine for COVID-19. Virus માટે કોઈ પણ medicines આપણ નથી જ પહેલાથી. પછી influenza હોય કે COVID virus હોય. We are just taking the painkillers. Of course, the interferon and other things are there, but some of these viruses, they are found to be polymeric. They are changing their shapes and they are not being trapped by any type of drugs. So, of course, the treatment is there with hydrochloroquine and hydroxychloroquine and remdesivir are prescribed, and they are found to be partially effective. So, this is particularly, we have gone through the immunology very briefly about the concept of immunology and what are the things which are providing with the defense mechanism in today's context. Historical aspects, we have seen that it was there, but it was supposed to be discovered properly. And once it has been discovered, today we are in a position to protect the individual body and to increase the lifespan of the individuals. So this is a particular basic concept or the aspect or the review aspect about the immunology in today's context. If you have any particularly specific questions, we can go through that.
Good afternoon, sir.
Good afternoon.
If you could elaborate on the role of vitamin D and E in boosting the immunity. How they directly or indirectly help in antibody formation?
See, vitamin D and vitamin E directly, they are not boosting the development or the production of antibodies, but actually they are undergoing for a supplements. It is a type of food supplements which is allowing a proper growth of the antibody producing cells. they requires the proper environment or the proper micronutrients and macronutrients to have the proper number or the properly developed B cells and T cells. See, B cells, they are converted into the plasma cells after the proliferation process, after the process of proliferation. Once the B cells are being sensitized or activated, they are enlarging. Small lymphocytes, they are converted into big lymphocytes. They are converted to plasma cells. And they are undergoing for multiplication or production of proteins inside the cell. And they are being released in the form of antibodies. During this phenomena, during this process, we require the vitamin E as well as what other you said, vitamin D, they are properly being required. If they are present, then you can boost this process of particularly the production of antibodies, or the cell activity can be properly being monitored. We are not actually monitoring, but this phenomena will go on in a proper way inside the body.
I would say.
Okay, sir. Thank you.
Sir.
Okay.
Hello, good evening, sir.
Good evening.
Sir, yesterday one sir came and he was talking about biotechnology, and at the same time he was talking about bioterrorism, and he was suspecting that the whatever we are suffering, that was done intentionally. But today you told that this particular COVID-19 is because of the while having different food in the form of rodents. Actually, we are not able to understand what it is. What do you say?
See, actually, it is you can say, we cannot call it at the level of microbiology or at the level of bioscience or the life science that it is a world level politics for suppressing somebody. But there are chances. See, here you can see that any particular chemical, it has the power of the production of antibodies, but it is not having the power to produce the disease. One statement I made that every pathogen is antigenic, but all the antigens, they may not be pathogenic. They are not pathogenic. They may be pathogenic. So here a particular composition or a particular makeup which may be totally foreign to the body. So here we are supposed to look that whether it can be prepared in the laboratory and later on it may be diffused into the environment. I have read some of the review articles regarding this also, that American scientists, they were working in the China and they have prepared this type of solutions, this type of preparations, and unfortunately or accidentally, they have spread it into the environment. So this theory or this hypothesis is already going on. Also, there is a hypothesis that the Chinese, they are using the rodents, bats particularly, or some of the animals for the eating purpose. So here you are disturbing the normal flora of those animals. Human beings, they are having a different normal flora. Human beings, they are not acquainted with these type of floras from the absurd animals or from totally different animals. We ourselves, we are not coming across the bat or some of the other animals. So once they are being disturbed, so dispersion of these type of pathogens or these microorganisms into the environment, there are chances. And they might have attacked over some of the immunocompromised or the immunosuppressed individuals, might have acquired that host properly proliferated, and that individual may be spreading that disease into the community. There are every chances. At present, we are not having any confirmation regarding this COVID virus, whether it has been arrived from the environment or whether it has been prepared from the laboratory or it has been escaped from the laboratory. We are not having an authentic document claiming that. We are not having.
Now, thank you very much, sir. One more question.
Sure.
Every day we are listening different news regarding adverse effect of vaccination. Do you feel vaccine is safe, or one should avoid it, or one should take it?
Avoiding is not the solution for combating the disease. We are hearing about the adverse effect, but 95% of the cases are found to be success. And regarding this 5%, we are not totally aware that those individuals who have taken the vaccines, they were immunosuppressed, they were having the diabetic, they were found to have some disease in advance, or they were already going from the blood pressure problems, or they were having some complications in advance. We are not totally aware of that. And science is the one of, you can say, that experimentation process, trial and error method. Of course, we are going for the clinical trials, and taking the vaccine is not a trial method. It is already proved method. But there are chances that the conversion or the adverting of the virus genes in some individuals, the probability, it has been reported. But 100% success, I have not seen today.
This vaccine is not advisable for those who are suffering from sugar-related disorders or blood pressures. You are not advising them?
No, I am not advising or particularly asking you for taking the vaccines, not taking the vaccines for the diabetic individuals or blood pressure individuals. But the government itself has given you the ruling or the guideline for the individuals for taking the vaccines and for not taking the vaccines. Here we cannot directly say that already who are suffering. See, there are various complications or various means of even the development of blood pressures. There are hypersensitive individuals. They are also suffering with the diabetes, or due to the cholesterol problem or due to some other problems also the individual is suffering with the blood pressure problems. So we have to go in detail. Of course, they are going in the clinical trials and they must have analyzed, they must have diagnosed that which type of individuals should not go for these type of vaccinations. They might have gone through that, but they have not given such type of ruling that only these individuals should take, they should not take. This is a total herd immunity process which is supposed to start. Whatever I think, the herd immunity process. Yes, the herd immunity process which was there in the polio vaccination or the polio campaign. This is one type of herd immunity development. And the 80% of the population is immune, then the rest of the 20% will enjoy. They will not suffer with the disease. This is a normal principle of immunology, that maximum population is resistant. Then other individuals, even though they have not gone for the vaccination, they may not have the attack of that virus, or the probability will be highly decreased. Normally, we used to teach in immunology. It has been mentioned in the books. So you can go in that way also.
Thank you very much, sir, for a very good explanation and a quite informative lecture. Thank you so much.
Good afternoon, sir. Can I ask you a question, sir?
Yeah.
Sir, it was a very good overview of the immunological science in the current scenario. I just had one quick question, that if I have already suffered from COVID, the memory of my immune system would be quite strong. So do I really need to take a vaccine?
No, it will memorize. The body itself, it is the basic principle that the immune response has the character of the memory. It will remember, of course. That is why we are going for the vaccination. Once you are vaccinated, the lifespan.
But if a person is already infected, does he really need a vaccine?
Yes. No, that is a different thing. That person should go for the vaccination or not. That we don't know. That we have to look what the government guideline is there. See, here the one big question is, this virus is found to be pleomorphic. It is not a single strain. 19 different strains. Yeah, there are different strains. And again, preparing the vaccines for all these different strains is a different thing.
Okay.
We are taking the DNA. Yeah, we are taking the DNA or RNA and we are synthesizing that protein. But these different strains, which has been coming out, which has been observed from different places or different individuals, or the virus once infecting the individual and being transmitted to other individual, the strain is found to be changed.
Okay.
So it is a big question. Vaccination should be there. Even those who have passed through the corona, they should go for the vaccination for the future security.
Okay, sir, and vitamin B complex, very few people recommend. I mean, which vitamin Bs are supposed to be good for this?
Vitamin B12 is a normally prescribed drug, which has been used for the treatment in most of the COVID hospitals.
Okay.
B12 ની ગોળીઓ તમને આપવામાં આવે છે, vitamin C ની ગોળીઓ આપવામાં આવે છે, and they are actually making the body healthy enough. Proper digestion. See, digestion, some of the basic criteria for keeping the body healthy. and they should be followed.
Okay.
If already we are having the complications, it should not be there. This is a normal view.
Okay. Thank you very much.